Hi everyone, I am Sam. Today we are going to talk about enzymes. Imagine for a moment that you're standing in a cozy kitchen on a quiet morning. The sunlight spills across the countertop and you're about to bake something delicious, maybe a loaf of bread. You carefully measure out flour, warm water, yeast, a pinch of sugar, and a bit of salt. You mix these ingredients and knead them into a dough. Then you let it rest. After some time, you notice something extraordinary. The dough puffs up, growing light and airy, as if filled with tiny balloons. If you've baked bread before, you know what's happening. The yeast, those tiny living organisms, is doing its magic. But what's really going on inside that sticky lump of dough? Inside that dough, quietly and without any fuss, a whole army of tiny invisible workers are hard at work. They're not people, not animals, not even complete cells. They're specialized protein molecules called enzymes, and they help the yeast break down sugars to release carbon dioxide. This carbon dioxide gas fills up the dough, making it rise. Without enzymes, that everyday kitchen miracle wouldn't happen. Now, imagine you don't know what enzymes are. Maybe you've never heard the word before, or if you have, you think it's some complicated scientific term reserved for textbooks and laboratories. But here's a secret. Enzymes are everywhere, quietly working behind the scenes. They're inside your body, helping you digest food so you can get energy from your breakfast. They're in the soil, helping bacteria break down dead leaves to release nutrients back into the earth. They're even harnessed by manufacturers to make cheese, wine, and countless other products. Simply put, enzymes are the hidden matchmakers that help specific reactions get going, reactions that might otherwise be too slow or might not happen at all. Before we dive deeper, let's turn back the clock to a time when no one knew about enzymes. Hundreds of years ago, people understood that certain materials could transform food. For instance, our ancestors knew that adding a piece of calf's stomach lining, rennet, would turn milk into cheese. But they didn't know why. To them, this was like magic, or maybe the will of nature. They didn't realize that a special substance inside the calf's stomach was speeding up the transformation of milk into curds and whey. They had stumbled upon an enzyme, but they didn't have the concept or the language to explain it. As human curiosity grew over the centuries, scientists began asking more detailed questions. What was it about yeast that made dough rise? What was it inside living things that allowed certain chemical reactions to happen so swiftly and reliably? While we now take for granted that chemical reactions can be guided by molecules, this was not always obvious. In the early days of modern science, some believed that only a vital force of life could trigger these transformations. Others began to suspect that something smaller, hidden, and distinct within organisms was responsible. In 1833, a significant step was taken when a French chemist named Anselme Payen and his assistant Jean-François Perceaux isolated a substance from malt barley that could break down starch into sugar. They called it diastase, the first enzyme to be discovered. This discovery was profound. Diastase turned starchy materials into simpler sugars, acting as a catalyst. Although Payen and Perceaux didn't yet fully understand what enzymes really were, nobody did at the time, their work lit a spark. Fast forward several decades. Scientists began discovering more substances that behaved like diastase, speeding up reactions without being consumed in the process. Each time they found something that could turn a specific compound into something else more quickly, they were seeing enzymes at work. Eventually, all these substances were grouped under a single term, enzymes, a word derived from the Greek en, meaning in, and zyme, meaning leaven or yeast, harking back to their discovery in yeast extracts. One lingering mystery remained. What exactly were enzymes made of? Were they mysterious, non-living chemicals that just happened to exist within living organisms, or were they something more organic? In the early 20th century, American scientist James Sumner made a groundbreaking discovery. He managed to purify an enzyme called urease and showed it was actually a protein, a large, complex molecule made of chains of amino acids. This was a critical turning point. If enzymes were proteins, then scientists could apply all the emerging knowledge of protein chemistry to understand how they worked. But understanding that enzymes were proteins was just one piece of the puzzle. Scientists still needed to figure out how these protein molecules could speed up reactions. After all, a protein is just a chain of amino acids folded into a complex shape. How did shape, of all things, turns some inert protein into a tiny chemical powerhouse. It turns out that shape matters a lot. Imagine a lock and a key. The lock has a very particular shape inside, and only a key with a matching shape can fit perfectly and open it. Similarly, enzymes have pockets or active sites on their surface. These active sites have a unique shape, a specially designed environment into which certain molecules, called substrates, fit perfectly. When a substrate locks into an enzyme's active site, it's a perfect snug fit, allowing the enzyme to twist and tweak the substrate molecule, weakening certain bonds or positioning certain atoms so that a reaction can happen much more easily. In other words, enzymes don't just speed up a reaction, they lower the barriers that would normally prevent the reaction from happening quickly. Think of a chemical reaction as a rolling ball. Without help, the ball might struggle to get over a high hill before it can roll down the other side. The enzyme works like a friendly crew that lowers the height of the hill, making it much easier for the ball to roll over. The reaction goes from taking ages to being almost instantaneous. Over time, scientists refined their understanding of this lock and key idea. They realized the enzyme wasn't just a passive lock waiting for a key. Instead, the enzyme's shape could shift slightly when the right substrate came along. This more nuanced model was called the induced fit theory. Picture it this way. When the substrate approaches, the enzyme subtly changes its shape, embracing the substrate more snugly, like a handshake that adjusts its grip. This fine-tuned interaction helps the reaction happen under gentle conditions, often at normal body temperatures and without harsh chemicals. If all this sounds complicated, don't worry. Underneath the fancy words, the concept is surprisingly simple. Enzymes are helpers that bring two puzzle pieces together and guide them to fit just right, so the puzzle can be solved in seconds rather than hours, days, or even years. Without enzymes, many reactions in your body would be so slow that life as we know it wouldn't be possible. Think of digestion, for example. We eat foods containing proteins, fats, and carbohydrates. Without enzymes, those large, complex molecules would be nearly impossible to break down into the simpler substances we need to stay alive. Enzymes like amylase in your saliva start working the moment you begin chewing, breaking down starches into sugars. In your stomach, pepsin breaks down proteins, and in your small intestine, lipases break down fats. By the time the food travels through your digestive system, enzymes have helped turn a complex meal into usable energy and building blocks for your body. This orchestration is happening all the time, without you giving it a second thought. Your cells are brimming with different enzymes, each dedicated to a specific type of reaction. Some enzymes help build larger molecules from smaller ones, allowing your body to grow and repair. Others help break large molecules down into smaller pieces, providing energy or raw materials. There are enzymes that help copy genetic material, ensuring every new cell has the right set of instructions. Without enzymes, none of these vital chemical processes would happen at the speed required for life. This idea extends far beyond the human body. Plants rely on enzymes to carry out photosynthesis, capturing sunlight and turning carbon dioxide and water into sugars and oxygen. Microorganisms in the soil use enzymes to break down dead plant matter, recycling nutrients so that new life can flourish. Enzymes are the chemical choreographers that keep our planet's web of life running smoothly. In human industry, enzymes have proven to be invaluable tools. We learned long ago that microorganisms and their enzymes can produce bread, beer, and cheese. Today, we use enzymes to achieve much more. Laundry detergents often contain enzymes that break down tough stains like grease, chocolate, or grass, allowing the detergent to do its work at lower temperatures and with less harsh chemicals. Enzymes are also used to clarify fruit juices, make paper bleaching more environmentally friendly, and produce biofuels. The magic of enzymes is that they offer a greener, more sustainable way to speed up chemical processes, often replacing harsh chemicals and high temperatures that were once needed. Stepping back, it's remarkable how the understanding of enzymes has grown from a vague notion that something in yeast and calf stomachs caused transformations, to a detailed and elegant theory about how tiny protein machines drive life's chemistry. But this journey of discovery was not straightforward. Early scientists struggled to convince others that non-living molecules could have such a precise effect on living processes. The debate over whether enzymes were living organisms themselves or just chemical substances raged for a long time. Once it was established that enzymes were proteins, the scientific community had to unravel the complexity of protein structure. No small feat, as proteins can be made of hundreds or thousands of amino acids folded into intricate three-dimensional shapes. Modern research on enzymes continues to push the boundaries. Scientists use advanced tools like X-ray crystallography and cryo-electron microscopy to visualize enzymes at the atomic level. They can see exactly how each amino acid lines up, how the active site cradles the substrate, and how subtle movements in the protein can change its activity. With this information, researchers can even engineer enzymes, tweaking their amino acids to make them more efficient, more stable, or capable of handling different substrates. This kind of enzyme engineering opens up a world of possibilities. We may be able to design enzymes that help break down plastic waste more efficiently, convert harmful pollutants into harmless compounds, or produce medicines and materials in more sustainable ways. Enzymes also play a huge role in medical research and healthcare. Certain diseases occur when enzymes don't work properly. For example, if you have a deficiency in the enzyme that breaks down lactose, the sugar in milk, you'll experience lactose intolerance, bloating, gas, and discomfort after eating dairy products. Understanding enzymes allows us to treat such conditions, either by adjusting diets, providing enzyme supplements, or developing therapies that correct the underlying problem. Enzymes are also critical in diagnostics. Many medical tests rely on enzymes to detect substances in the blood. For instance, glucose meters, which help people with diabetes monitor their blood sugar, use an enzyme that reacts with glucose to produce a measurable signal. Without enzymes, many of the rapid and easy diagnostic tests we rely on wouldn't exist. The complexity and versatility of enzymes reflect the complexity and versatility of life itself. Think about it. All life forms, bacteria, fungi, plants, animals, rely on enzymes. Despite the enormous variety of life on Earth, the fundamental chemistry that supports it is based on the same principle, using enzymes to speed up and direct the chemical reactions necessary for survival. In this way, enzymes are a unifying thread across all species, connecting the smallest microbe in the soil to the largest whale in the ocean. And yet, for all their importance, we rarely think about enzymes. We eat, breathe, and grow without consciously thanking them. We wash our clothes, brew our coffee, and make our bread rise without acknowledging the tiny protein catalysts that make these tasks easier. Perhaps that's one of the reasons why learning about enzymes can feel so profound. It's like discovering a hidden dimension of everyday life, one that's always been there, silently at work, ensuring that everything runs smoothly. If you picture your body as a bustling city, each cell is a neighborhood, and each enzyme is a skilled worker in a specific factory. The city hums along because every worker knows their job and performs it efficiently. If an enzyme is absent or malfunctioning, the entire neighborhood might grind to a halt, and problems can arise. When everything is working as intended, you never even notice the complexity. Enzymes keep the traffic moving, the lights on, and the goods flowing without calling attention to themselves. Beyond the world of living organisms, the concept of enzymes teaches us something important about chemistry and nature. That not all chemical reactions are brutish battles of heat and force. Enzymes show us chemistry can be gentle, precise, and elegant. They demonstrate that finely tuned structures can guide molecules through complicated dances with minimal waste and effort. In this sense, enzymes inspire scientists and engineers to design better chemical processes, ones that are kinder to the environment and more efficient. As we think about the future, enzymes may hold the key to many problems. Climate change, resource scarcity, and pollution challenge us to find more sustainable ways of living. Enzymes offer a blueprint for doing just that. By studying them, understanding how they work, and learning to engineer them, we can develop techniques that reduce our reliance on harmful chemicals and extreme conditions. This is not just wishful thinking. Researchers are already engineering enzymes to break down plastic, turning a vast environmental problem into manageable parts. Others are working on enzymes that help convert agricultural waste into biofuels, reducing our dependence on fossil fuels. So, by now, we've traveled through history, starting from the early days when no one knew what enzymes were, to the discoveries of diastase and the identification that enzymes are proteins, to the lock and key and induced fit models, and finally to modern enzyme engineering and their applications. All this from the quiet miracle happening in a loaf of rising bread. The next time you enjoy a slice of bread, sip some wine, or peel an orange, remember that enzymes had a hand in making those experiences possible. Consider that enzymes are essential not just in the kitchen, but also inside you, inside every living creature, and inside countless industrial processes that shape our modern world. They are nature's nanomachines, perfectly crafted to make life's chemistry possible. Understanding enzymes might feel like gazing into the innermost workings of life, peering behind the scenes at the small, precise interactions that give rise to big, wondrous phenomena. It's a reminder that the world is full of complexity, hidden in the simplest things. Something as ordinary as rising dough can become a window into the fundamental chemistry that underpins existence. In the end, enzymes teach us that life isn't just about big events or visible changes. It's also about the countless tiny actions happening every second, each one guided by a molecular helper, ensuring that the grand tapestry of life remains intact and vibrant. By learning about enzymes, we gain a deeper appreciation for the delicate orchestration playing out both inside our bodies and across the natural world, every moment of every day. That's all for today, and I'll see you in the next episode.